Good afternoon, everybody. And I would like to thank SSBMT and SSH for giving me the chance to present my work here today. So my presentation is a little bit, as you see, is a little bit different from whatever we have been hearing since yesterday. It is a pure. So it is a little bit different from whatever we have been hearing since yesterday. It is mostly lab-based study, the basic study, the basic science study. So I hope that you enjoy it. Thank you. So the topic of my talk is the consequences of interaction between placental mesenchymal stem cells and the platelets. So I will give some background information followed by introduction of the topic, and then I will discuss about the objectives, results, and finally the conclusions. Mesenchymal stem cells. We all know that mesenchymal stem cells are MSCs. They are multipotent adult stem cells, which have the property to differentiate into adipocytes, chondrocytes, and osteocytes. They can differentiate also into other stem cells that include the neurons, cardiac cells, muscle cells, and other cell types. So they are isolated almost from all other adult tissues, but the most common ones are adipose, dental polyp, bone marrow, umbilical cord, as well as the placenta. So MSCs possess immunomodulatory properties. They have immunosuppressive properties. They have differentiation potential. They have tissue regeneration potential, and they have the ability to home to the sites of inflammation. And because of those properties, they are used in cell-based therapies, including cancer, cardiovascular diseases, immunological disorders, and diabetes, etc. So in cell-based therapies, they have been used as intact cells for therapeutic purposes, or their cellular product have also been used for therapeutic purposes that include the exosomes or conditioning media we also call a secretome. So although they are the excellent sources for cell-based therapy, but still in humans and in animal studies, rare and minor adverse events have also been documented in MSC applications. So in animals, we have seen increased death rates, which have been seen because of the thromboembolic events after infusion of these MSCs. These thromboembolic events may occur through different mechanisms. They include the size of the cell, size of some of MSCs, which may get trapped and occlude small vessels in lung capillaries. And also, the other mechanism is that some of these MSCs, they promote coagulation through high expression of procoagulant molecules, which trigger the clotting cascade. So we have been working on, for a while, on placenta-derived stem cells. From now, I call them pMSCs. So we have isolated and characterized 3 types of pMSCs from human term placenta. That includes the DBMSCs, or deciduo basalis mesenchymal stem cells. They are isolated from the maternal region of human term placenta. DPMSCs are deciduo paratilis. That is also from the junction of maternal and fetal side of human term placenta, and also chorionic villus MSCs, or CVMSCs. They are isolated from the fetal region or villus region of human term placenta. So why placenta? Why did I choose placenta for isolation of MSCs? So because placenta, as all of us know, is a waste product, it is discarded eventually after the birth of the baby. So although we have to get the informed consent from the patients, but still it is generally devoid of major ethical considerations. And unlike other tissue collection, these we don't have to use any invasive procedures. So we have to use the tissue is collected from non-invasive procedures. And it is a rich source of stem cells, and we get substantial number of placenta from a small piece of tissue. And also, as like other MSCs, pMSCs are immune modulators. They have the broader differentiation potential, and they have limited proliferative potential because we can grow them only up to maybe 5 or 6 passages. After that, they either differentiate, shed, or die. So they grow on plastic also, and because of their limited proliferative potential, they have low risk of teratoma formation post-transplantation. And as compared to other MSCs, like bone marrow MSCs, they are smaller in size, therefore no occlusion or little occlusion was found in lung vessels in animal models. So we undertook this study because of the immunomodulatory properties. They make them excellent sources for cell-based therapy against immune-mediated diseases. And they also secrete soluble factors that exert immune responses on multiple cell lineages like T cells, B cells, NK cells, monocytes, etc. So we hypothesized that they may interact with platelets also and modulate their functions, and in turn play a role in atherosclerosis and thrombosis phenotypes. So the aims of this study was to see the effect of pMSCs on platelet activation in presence and absence of various agonists, and to investigate if pMSCs modulate platelet functions, playing a role in platelet-mediated thrombosis and atherosclerosis. So we isolated platelets, we washed them and treated them with the cellular component as well as the secretome of pMSCs. The cellular component was used as different ratios as well as the secretome at different concentrations. So platelets were stimulated using different agonists after treatment with the pMSCs and their secretome, and we used different agonists, including collagen, ADP, as well as thromboxane A2 inhibitor U46619. And the effects of pMSCs and their spinnergens on platelets was evaluated by flow cytometry for the expression of activation markers and by platelet functional analysis. So we used human HUVEX as the control. So just a small just a recap of the platelet activation and functions. Usually the agonists, they include thrombin, ADP, CRP, whatever is it. So they bind to the receptors on the cellular surface, on the expressed on the platelets. And this binding initiates an signaling cascade that on one side results in selection, the surface expression of p-selectin, and on the other side it activates the otherwise inactive integrin that's called as alpha-2b beta-3. So this is called as inside-out signaling. So this activated integrin alpha-2b beta-3 is recognized by an antibody that's called as PAC1. So this fibrinogen now binds to the activated form of integrin alpha-2b beta-3 and initiates the outside-in signaling that results in granule secretion, followed by amplification of platelet activation, stable adhesion, and spreading and aggregation, etc. So the platelets treated with different concentrations of conditioned media from DB-MSCs at 5%, 10%, and 20% showed this, the dose-dependent decrease in p-selectin expression as compared to the untreated control. Here, the 10% and 20% treatment show significant decrease in expression as compared to 5%. The resting one did not, we didn't treat the resting ones. These are the resting platelets without any treatment. So we looked at the PAC1 expression also. It also showed a similar trend where 10% and 20% showed decrease in PAC1 expression as compared to 5% and the untreated controls. Similar trend was was obtained with the DP-MSCs from deciduous paratilis MSC conditioned media at 10% and 20%, where we can see the decrease in 5% also. There was significant decrease in PAC1 expression as compared to the controls, whereas in the PAC1 expression also showed decrease in the dose-dependent manner. However, for chorionic villus MSCs, we interestingly did not see any change here. We did not see any change here. There was no change in PAC1 expression after treatment with this conditioned media at 5%, 10%, and 20% after incubation with collagen at 2.5%, 2.5 microgram per ml. But as compared to untreated control, we did not see any change here. And similar trend was found in the PAC1 expression. Also, no change was found in the PAC1 expression. So next we looked at different ratios of the cellular component. This was the conditioned media. So now we look at cellular component at different cellular as 1:1, 1:2, and 1:5 cellular ratios. And in DB-MSCs showed P-selectin levels decreasing significantly at 1:2 and 1:5 cellular ratios. And this is a resting one where we didn't see any change. Definitely, and in the DP-MSC treated, the cellular component treated platelets, we also see the change, the decrease in P-selectin levels at 1:1 and 1:5 cellular ratios. But as observed before, we did not see any much change. Although there is subtle decrease in P-selectin expression here, but it was not significantly, it was not statistically significant. So we looked at the PAC1 expression also of pMSC treated platelets, and as you see here, that DB-MSCs showed a significant change, a significant decrease in PAC1 expression. DB-MSCs also show significant decrease, whereas CV-MSCs did not show any change. So these are the resting platelets where we didn't see any change. So next, we looked at the platelet functions. That the first experiment that we did is platelet spreading on immobilized fibrinogen that was on this what's called glass coated cover slips. We put the platelets treated with different pMSCs as well as with agonists, and the unwashed, the unbound platelets were washed, and then the bound platelets were fixed and looked under the microscope. So we took as you can see here, this is untreated control, and we give the time points for 15 minutes and 30 minutes, and this is the treatment here. So when we quantify this and to look at the average surface area of the bound platelets, to look at the surface area of the spreading, so we see that here that DB-MSCs and DP-MSCs again here that decreases the surface area as compared to the untreated control. Although in CV-MSCs also they show decrease in the average surface area, but it was not significant between the two time points that we see here. After that, we looked at the adhesion. Adhesion is also like, it is a calorimetric assay where we see the binding and the adhesion of the platelets to the fibrinogen, to the immobilized fibrinogen in a 96-well plate. That's how we do, and we do the calorimetric assay where we look for the acid phosphatase release of the adhered platelets. So this is, as you can see here, there is a percent adhesion at 30 minutes and 60 minutes. Although in DB-MSCs, we see significant change at 60 minutes. Although in 30 minutes also, we see there's an early event, we can see the change, but it is not significantly significant. But in DP-MSCs, as we can see that it is statistically significant at 30 minutes as well as at 60 minutes time for adhesion.
Although in CV-MSCs, we see some change, but it was not statistically significant. So after that, we looked at the aggregation studies of these treated platelets. So as you can see here, this aggregation was done, as we all know, this is just the electrical impedance that depends upon the turbidity of the sample. So it gives us the tracing in the machine. So because of the turbidity, as you can see here, the untreated control and CV-MSCs show increased aggregation after 5 minutes, and as compared to the DP-MSCs and DB-MSCs, which show lower aggregation potential and lower aggregation the tracings here. So this was quantified. And you can see that DB-MSCs and DP-MSCs show decreased, significant decrease in aggregation, in total aggregation after 10 minutes. That is the total time that we take it for aggregation as compared to the untranslated controls. So after that, we did the mRNA analysis and looked at a couple of prothrombotic and antithrombotic genes that were expressed in these stem cells, or the pMSCs. As shown here, we looked at antithrombotic genes like TFPA1 tissue factor pathway analysis inhibitor 1, protein C1, protein S1, serpin C, and thrombospondin 4. As you can see that they show higher expression in DB-MSCs and DP-MSCs. All of them are highly expressed here, except the serpin, but they are highly expressed and statistically significant. So looking at the prothrombotic genes like VWF, GP6, phospholipase C, gamma, GRK5, and plextrin, we see that they are definitely lower, showing low expression as compared to the HUVEX, but interestingly, the CV-MSCs showed higher expression of VWF as well as phospholipase C gamma as well as GRK5. So this expression, this mRNA expression, was further verified by the protein levels of this prothrombotic and antithrombotic genes, and the upper panel shows the protein bands, and lower panel shows the densitometry analysis of those bands. And this data corroborates whatever we get in the mRNA analysis. So platelet activation and atherosclerosis. So what is the relationship between platelet activation and atherosclerosis? Platelet activation and hyperactivity, we all know that contributes towards pathogenesis of acute thrombotic disorders. So they play a central role in inflammation upon their interaction with leukocytes. And in addition, activated platelets also interact with lipids. It has been linked to thrombotic risk. So oxidized LDL oxidized LDL, or oxidation of LDL, promotes atherosclerosis. We all know that through inflammatory and immunologic mechanisms that lead to formation of macrophage foam cells. And we have CD36, it is glycoprotein 4. It is a multi-ligand receptor that is expressed in macrophages, dendritic cells, and also in platelets. So these CD36, it recognizes certain oxidized phospholipids. So in platelets, CD36 binding leads to oxidized LDL that results in activation of specific signaling pathway that induces platelet hyperactivation. And this CD36 binding to oxidized LDL is believed to contribute the procoagulant state that is associated with hyperlipidemia and contribute to atherosclerosis. So we look at using the commercially available oxidized LDL kit. We did aggregation studies for all these pMSC treated platelets. And as you see here, As you see here, that the collagen plus oxidized LDL traces show high aggregation here, as well as we expected with collagen. But when they were treated with DB-MSCs, either the conditioned media or the cells, this aggregation goes down. And that is what we have earlier found, that collagen plus DB-MSCs showed lower aggregation of these platelets. And the quantification here shows that collagen plus oxidized LDL shows higher expression of higher aggregation here as compared to collagen only. But when it was treated with DB-MSCs, so this significantly reduced the aggregation of platelets.
1 minute, doctor.
So so to conclude this, we show that a subset of pMSCs reduced activation of platelets, both via the direct contact as well as with secretome treatments. So decrease in platelet activation translated into diminished spreading, limited adhesion, and minimized aggregation. pMSCs also decreased oxidized LDL-induced CD36-mediated activation, establishing their protective role in atherosclerosis. And although pMSCs, they secrete this pro and they differentially express pro and antithrombotic proteins, which might be responsible for the activation of agonist-induced platelet functions, but we do not do any studies, so we don't know which proteins are responsible and what is the mechanism behind that. So these data suggest that pMSCs can be safely used for any disease setup, and we might not see any thrombosis or atherosclerosis phenotypes in those patients given those pMSCs. So I would like to thank my colleagues at KMARC. This is this cell therapy, this is stem cell and regenerative medicine at Cell Therapy and Cancer Research team. This is Dr. Yasser, Abdullah, Ireem, Dana. They are my technologists who conducted this study, and also this research proposal and processing department. They approved this study, and KMARC Research Funding Committee for funding this grant RC15. And KMARC operations for their logistical support, and also at Baylor College of Medicine, I am thankful to Dr. Subhashri Pradhan for conducting all the aggregation studies. Thank you very much.
Thank you, doctor.